started. Welcome to the 2020 Senior Showcase. Hooray, hooray.
Yay.
Yay.
Yeah, thank you. Excellent. You are being hosted here by, I want you all to pay attention to who's here. Alan Sells is over here, producer. He's to my left. I don't know where he is on your screen. He's making everything happen from a video standpoint. For those of you who watched your 360-degree video today, a virtual tour that brought you back into the Bethel, that was thanks to Dr. Marshall, who placed things all around the Bethel. And Pam, she put the things in the mail and made sure that we found everyone, got them all the right stuff. So you have these people here to thank, and you have marketing and communications as well to thank for the virtual reality goggles, the video, and for a lot of the support that's going on here. But we're really delighted that you're here, and we'd like to kick off our showcase with a very special welcome. Alan.
Honors Class of 2020. I know this is not how you envisioned your senior year being. However, you made adjustments, you were successful, and as a result of this experience, I have no doubts that you will be stronger because of it. When you entered the honors program, it was a class of 150. Now it's an honors college consisting of 350 students. During this time, you've offered Dean Cook insight and feedback, helped the college change the admissions process, and engaged extensively in study abroad and global experiences. Certainly your research had to be changed, and I believe you all did that in a very good fashion. Most of you imagined being able to work in a laboratory or face-to-face interactions with colleagues, which certainly was not possible. You did complete these, however. During this time, your research has spanned the globe, and now you're off to different parts of the world. Congratulations on graduating and now becoming an alumnus of the University of South Alabama. Please stay in touch with us.
It's my pleasure to welcome everyone to the 2020 showcase for this year's graduates of the Honors College at South Alabama. As you know very well, this year's event is being delivered virtually rather than in person because of the unusual circumstances facing the whole world right now. This is a challenging time for all of us, and I certainly regret that this year's seniors are not able to return to campus to enjoy all the many graduation ceremonies, including this one. And even though I have these regrets, I'm also grateful for many things, including the fact that each of you was able to finish all of the requirements to earn your degree with honors. I'm extremely proud of the Honors College at South Alabama. I know that every student who completes all of the rigorous requirements of the Honors College has received an outstanding educational experience that rivals that of any college or university in the world. This is demonstrated by the outstanding accomplishments of this year's graduating class. The class of 2020 includes 35 Honors College graduates and 9 departmental honors graduates. They represent many majors, ranging from communication to biomedical sciences and political science to engineering. One student won a Hollings Award. Another was a recipient of a National Science Foundation Research Abroad Scholarship. We have an NSF REU Award winner, and we have an NSF Fellowship Honorable Mention, among many other students who've won outstanding awards among this year's class. Over the past month, each graduate has completed a thesis project, presented it virtually, of course, on topics ranging from palliative care, marketing in higher education, and the relationship between cognition and reading skills. As part of their studies, the graduates have traveled all around the world, including visits to the United Kingdom, Poland, Spain, Brazil, the Czech Republic, and many other destinations. And each of you has persevered through a difficult final semester interrupted by a pandemic. I could not be more proud of each of you for all that you have accomplished, especially under these difficult circumstances. I'd also like to pay tribute to the faculty who have worked with these students during their course of studies at South Alabama, but especially I'd like to thank the faculty mentors who guided you through your senior research project. These faculty did so as extra duties on top of their normal jobs, and this year they did it under extraordinary circumstances. So I'd like to thank all of the faculty who helped participate in guiding these students through their experiences here at the Honors College at South Alabama. So to the graduates, congratulations on all of your accomplishments. You worked hard, you persevered, and you've accomplished extraordinary things. And we look forward to seeing all that you accomplish in the years ahead. Congratulations.
Hi, folks. Hope everyone is doing well. Again, we're sorry that this is the format. We had really wanted a lot more, but I want to say to start off with, we've really enjoyed having you guys the last 4 years. I remember all of y'all coming in. I remember the retreat. I remember being on the ropes course with some of you, sometimes a little too close to some of you, perhaps. But we had a great time, and I can't believe it's been 4 years since y'all came. Obviously, because you've been part of the college all this time, you understand how big an endeavor this is. You've seen the way that myself, especially Dr. Cook, Mr. Sells, Pam, everybody has worked so hard. And the reason we do it is for this moment, to be able to send you guys out into the world with a unique set of skills, a unique perspective on the world, and hopefully a better experience. But again, I want to underscore that there's a lot of work that goes on here, and it begins at the top. You don't have an honors college like this without proper support of the university. As you just saw, you just heard from President Walder, you just heard from Provost Johnson. Without their support and their real care about what happens to this college, you could not have had the experience you had, and we could not do the things for you that we're trying to do. So I want you to remember them. Also, again, we always have to thank the USA Foundation. It has been such a supporter of the college, and before that, the program for years. Again, what that makes possible for all of us is truly remarkable. But as you know, there's lots of other people. Of course, you know the four of us. They're kind of around the Bethel all the time. But let's not forget the people that really are special for you guys, that work so hard. For example, the people in scholarships. Dr. Forbes, Dr. Hamilton, Dr. Stedman. Let's not forget the Honors Fellows, Drs. Guzzi and Jackson. Let's not forget Dr. Wheeler West over in undergraduate research, who so many of you have worked with so far. But to echo what Dr. Johnson said, I think there's nobody that we really need to owe our thanks to more than your mentors. Your mentors have made space in their schedules. They made space in their lives for you guys, and we all owe them such a debt of gratitude. Those of us that work for the college do, the university does, you guys do, and I hope you take the time to let them know. But I also want to point out there is honors faculty more general. We try to pick the best teachers on this campus for you guys. We try to have people that are going to expand your mind and give you new ideas. So I really hope, again, that you recognize how many people have worked to make this possible. But at the end, I want to also thank you for joining us. I want to thank you for taking the chance to join our program, realizing there might be something here for you, for sharing your experiences, for letting us work with you, and also a shout out to your parents for raising such capable kids. We're really impressed by you guys. We really are. As you know, by the way, because this is a kind of a rough transition, but as you know, one of the things that we really are big about here at the college is the creation of culture, which we often call research. But again, as you know by now, research might be too limiting a term. So to underscore how important this is, one of the things you got in the email that we sent you was a poster session, a virtual poster session of the research that all of you have done. So I really encourage you, if you haven't kept up with somebody that you came in with, go through there and see what they did. It's all impressive stuff. I know that all of Mr. Sells, myself, Dr. Cook have been attending defenses for several weeks now. And it's a lot of time, it's a lot of effort, sometimes to read, to try to understand things that frankly are outside our discipline. But it is a hoot. We wouldn't trade it for the world. It is so much fun to see what you've done and to remember what you look like on that ropes course and then see what you've become. It's just great. So to underscore that, as I said, look at the poster session, but to particularly underscore it, we have a couple of students who have taken the opportunity to set up video presentations of their research. And so I invite you to just hang out for a few minutes and watch presentations first by Christian Manganti and then Bailey Manning, telling you a little bit more about what they did, just in case you need a little bit more flavor for it. Because I know when you're doing your project, you got your head down, you're focused on yourself. Let's look at what some of your colleagues have done.
All right, hey everybody. So this is Christian, and it really sucks that I have to come at you through either your headphones or your computer speaker. I really hoped that we would be able to do this in person because it's been like way too long since we've all seen each other, but it's the best we got. So today, if you want to stick it out throughout this whole recording, you're going to get a really broad overview of what my project was. And the good news is the BMD department's already heard this, so I get to make it a lot more accessible for you interdisciplinary crowd. And so, in the simplest of terms, my project involved using gastric lavage fluid, which is a patient sample, to detect pancreatic cancer. So, kind of starting off with the obligatory background, pancreatic cancer, or pancreatic ductal adenocarcinoma, or PDAC, is one of the scariest cancers, with a lot of the general public kind of like equating diagnosis with a death sentence. And to put that in numbers, only 21% of these patients live 1 year after being diagnosed, and that number drops by more than half to like 9% of patients who live 5 years after being diagnosed. And so depending on what article you consult, this long-term survivability number usually ranges from like 2% to 10%. And so all of this considered, it's easy to understand why PDAC is in the top 5 of all cancer deaths. And so it's in that top 5, not just because these tumors have high metastatic capability, but also because PDAC is unique in that it has this self-fulfilling prophecy that kind of plagues especially gastric cancer specialists. And so PDAC is a disease that's asymptomatic in its early stages. And that might sound good, but what that really means is these patients have their growths that grow and progress without them ever noticing until the later stages where the disease is more advanced and more aggressive. That's when it's usually caught, just because they actually notice it now. Now, unfortunately, since we catch it in those later stages, these patients don't really have a lot of time to acclimate to treatment. And so in that window, there's not really enough time for researchers to study the disease progression in those patients. And so we unfortunately have to keep saying that there's no real hallmarks of early disease, and so PDAC is unfortunately still asymptomatic as far as we know. Now, this cycle is kind of the main issue that cancer diagnostics tries to solve. And so these diagnostics would be most effective if they broke the cycle right here. Now, our best weapon for doing that is biomarker research, which involves trying to figure out exactly what a cancer looks like on a molecular or cellular level. And so these researchers might ask, what proteins are modified in this cancer that give us a very specific way to track it? So one example that you guys probably heard of from like biology class or the commercials is the HER2 hormone receptor, and that's found on the surface of cells. And this biomarker is used to delineate between certain types of breast cancer. Now, the good thing about biomarker research is that it yields very effective diagnostics, but it often takes years to figure out what those biomarkers are and to produce a test that can reliably screen for them. So I've kind of jokingly dubbed my project the solution to the slow solution because it completely circumvents the slowness of biomarker research, and it does so actually by using very simple, very common laboratory techniques, these two that you see here. So speaking of those two, this is probably going to kind of sound like I'm getting in the weeds here, but I'm essentially going to give you like a behind-the-scenes tour of how this works. And so the beauty of this diagnostic test for pancreatic cancer is that we've devised something that still differentiates between cancer and other things. You know, that's exactly what it's supposed to do. But we do so based on not specific biomarkers, but based on these very generalized patterns in immune reactivity. And so immune, like directly referring to immune system. So if you guys remember that this assay uses gastric lavage fluid, this GLF is a liquid sample that's obtained from patients through cleaning their colons in preparation for a colonoscopy. And so since it's derived from that GI tract, it's got all the digestive enzymes in your gut, you know, the stuff that eats up your steak. but it's also got IgA class antibodies. And so these antibodies are sensitized to different molecules, just like all antibodies. And when they are, they would readily bind to those molecules if they encountered them. And so that's an essential step in a general immune response, when you get an infection. Now, one of the ways we can test what antibodies will bind to is using a peptide array. And so this peptide array is this big grid of peptides with like thousands of them. And those antibody samples are applied to these arrays, and we can see which peptides those antibodies bind to via a color change. And so these two pictures that are on the left half of the slide here, these are just hypothetical data that I kind of pulled from the internet to show you guys how we use patterns in immune reactivity to make a diagnostic test. So if we pretend that these two pictures are two copies of the same array, one treated with a pancreatic cancer sample and one treated with a normal healthy patient sample. And so I won't go into too much detail, but basically the fact that these two pictures are different, like different colors in different places, that tells us that PDAC patient IgA and normal healthy patient IgA act differently. And so that difference, and that difference alone, forms the basis for this diagnostic test, not a specific biomarker. And so that's more qualitative, obviously, with the two differences in color. But this sort of binding reactivity can be quantified using an enzyme-linked immunosorbent assay, which is essentially the forming of these molecular chains built out of defined reagents. And so you see, I know what each of these pieces are. Now, I won't go into too much detail again, but based on this ELISA and how we've constructed it, if a patient has PDAC, we should get high numbers, and if a patient does not, we should get low numbers. Pretty simple, right? All right, that was kind of zoomed in, but I'm going to zoom out for just a second and clarify that the grand goal of this project was to produce a diagnostic test for pancreatic cancer that we could use to screen for this cancer across a population. And so this is what that development process looks like, kind of condensed and to the point. So you might get a method and start getting some successful data, and then you want to replicate that on your own to make sure that your method is sound before you pursue more, we'll call them official measures like clinical trials, intellectual property, and hopefully when you're doing that, some big shot biotech company will pick up on what you're doing and they'll offer to develop that into a product and commercialize it for you. And so everyone really wins in the end. And so I'm going to reiterate one more time that this is exactly what it should look like in an ideal world. But this is what my development process looked like. So we're seeing a lot more twists and turns here, and it actually turned out to be kind of a really fun project. And so let me kind of clarify once more that throughout all of these steps, what we were doing is testing the effectiveness of our diagnostic method with given patient samples. And so we run the method, check the data against what we know about the diagnosis of each patient, and so we can gain how effective our method is at actually identifying pancreatic cancer and not identifying other things. So I'm not going to hit data from all these steps, but I will kind of hit all the important parts with you. And so this is some trial one data here, and what we do is we gather the data for each sample and subject it to this self-designed analysis process that I called the ratio of reactivity, or ROR. And so in trial one, the good news is that we ended up getting data that almost perfectly matched the patient diagnoses we were given. And so in the left table, we have our cancer group, or all the patients that had cancerous growths, and they had very high ROR. So we have 33, 17, 112. Compared to the right table, where we have all the other patients. So those who were healthy, those who had minor issues that weren't cancer. And so we see that they had very low RORs. And so biggest number over here is, I think, 8 or 7.7, around there somewhere. And so that's exactly what we should have been seeing. That's diagnostic merit right there. We get high numbers for cancer patients, low numbers for normal patients. And so that's exactly what we want. And in fact, when I ran a little 2-sample t-test here, we get that the cancer group RORs are significantly higher than the normal group RORs. And so that's a little bit of statistical reinforcement to what we qualitatively see. Now, here's the fun part I hate confessing. When we repeated this method twice more using the same patient samples, our data never quite looked the same. And so what alarmed us specifically is that if you remember how the cancer samples in the trial 1 data I just showed you, those had really high RORs. But in trials 2 and 3, they had super low RORs. And so that's not good, because essentially that's a false negative. We have a test that's telling us that a PDAC patient is healthy, which is wrong. Now, to kind of track what might be happening or why that might be, I compiled some data from a single patient sample CLNSN 1221, which is a normal patient sample. And now, what we're thinking, if you guys pay attention to this conditions column right here, what we're thinking might have affected, my data is the usage of the sample in each trial. And so, assuming that everything else was replicated perfectly, that difference in sample usage should have been the reason that the data looked different. And so throughout the different trials, what we're thinking is happening is the IgA in those samples is degrading because of how I'm using them. And so that would naturally mess up the data, because the data is dependent on IgA. All right, guys, kind of cliff notes all of my discussions and conclusions with you guys. So we can't confirm this method's reliability, unfortunately, because the data was not reproducible between trials. And so this was especially alarming because we actually pursued other aspects to confirm the reliability of this assay, and those worked out perfectly. And so we're not really sure what might have happened in between trials there. Now, one time when I was presenting this maybe about a year ago, this really smart girl over at MCI approached me after a presentation and commented on the idea that maybe this method isn't true early detection because of the availability of GLF. So in short, if you guys remember that GLF is obtained from patients before they get colonoscopies, those patients who get routine colonoscopies are usually middle-aged or older. And so what that says is our assay is reliant on using a sample that's obtained from older patients who would likely have more advanced pancreatic cancer. And so there's an issue with us calling this an early diagnostic test. So one solution to that could be using human serum, which is derived from blood, as the sample instead of GLF. And so in theory, that sample should have the same patterns in immune reactivity. Now, of course, all patients of all ages really get blood work done, so the screening capabilities are much higher if we were able to switch the patient sample that we're using. Now, even with those issues, I kind of still have to reiterate how I really believe that those trial 1 results were cool. They were great. Considering that statistically significant difference, the way the RORs matched patient diagnoses, and the way that we achieved that data even with our small sample size. But if I were to pick one thing that surprised me really, it would be these 2 samples here. Now, these 2 samples were not true PDAC samples, but they gave us higher numbers than the actual PDAC samples did. And so you say to me, Christian, you told me this was a pancreatic cancer test. Why are things that aren't pancreatic cancer testing positive? And I'll say to you, I mean, there's no real way for us to know why the test is sensitive to other gastric cancers, but that's not necessarily an issue. It's actually pretty great because that means that this test could maybe be used as like a first line of defense screening method against all gastric cancers or abnormalities. And so if your physician saw that your numbers were high, she or he then might be able to recommend more reliable diagnostic methods like imaging. Now, unfortunately, and hate to admit this too, all of this has pretty much been laid to rest because our research funding got cut and our access to these samples has ended due to some personnel changes over at MCI. All right, guys, so that's all I got for you today. I hope it wasn't too bad, and I hope you got the gist of how cool this project is, or could have been, if we were able to continue it. Now, for this project, I first have to thank Dr. Duran, and I think we all do, really. For me, I appreciate him so much because he actually set me up with this gig at MCI, where I've been for the past 3 years, and he did it without knowing me really well and for trusting me for some reason. Now, I also have to thank Steve McClellan, who was the man that Dr. Duran introduced me to, who became my mentor for the past 3 years. And I really have to thank him for being so accommodating and welcoming at MCI, you know, as well as providing me with such great support and really all the work that I cared to do. I also have to thank my thesis committee, Dr. Rice, who's the new chair of the Biomedical Sciences Department, as well as Dr. Swiger. And so I know that we were all terrified of the thesis project for, I don't know, the past 3 years. And my committee definitely eased my fears quite abruptly, as we started meeting last fall. Along that same vein, we have Dr. Mockett, who serves as the kind of BMD coordinator for the honors thesis projects. And so not only did he teach a solid like 1/3 of my BMD curriculum, but also he was very meticulous in planning and instructing the 499 courses and really laying out his expectations very clearly. So speaking of clear expectations, we also have Dr. Hamilton, who wasn't at my thesis defense, but who I hope might hear this. And so if you took her for Honors 301, you also have to understand how helpful and encouraging it was when she really procedurally laid out clearly what the requirements were for the prospectus, the thesis, and the defense, and that's definitely helpful. Kind of turned the thesis process, from like a scary ghost to like a scary ghost who we understood. We, of course, have to thank everyone at the Honors College who support us and encourage us so greatly. And lastly, I'd like to thank whoever's listening, not just because you sat through my babbling, but also because I've always considered the Honors College my family. And we aren't all extremely close, obviously, but it always meant a lot to me that everyone in our honors class, despite not spending every day together, I could see any of you just randomly, and we could pick back up a conversation like we didn't just spend 3 months or 2 years apart. And that says a lot about how much we support each other, and each of you really means a lot to me, probably more than you even know, or more than I mean to you, but that's okay. But anyways, thanks guys for listening, and that was my thesis project.
Thank you, Christian. As you can see, I'm using the little reaction things. If anybody wants to clap, or you can turn on your microphone and clap for a few minutes if you want to, or a few seconds, I should say. Thank you, Christian. Lots of clapping, I see. Also, I'd like to invite you to add questions to the chat. We have another presentation coming by Bailey Manning, and that'll start in just a second, but I'd like to encourage you to think about adding questions to the chat. You also can add questions about the posters that you saw to the chat. This is one way for us to be interactive. And then after the presentation, we will continue with another portion of the showcase that you'll also, I'm sure, very much look forward to. But now, without further ado, our second presentation by Bailey Manning.
Hello, my name is Bailey Manning, and the title of my project is Evaluating the Role of the Base Modifying Enzyme APOBEC3B in Cancer Cells with DNA Repair Defects. The APOBEC family constitutes a group of proteins that induce DNA sequence alterations by deamination of deoxycytidines to deoxyuridines. In simpler terms, deamination occurs when APOBEC catalyzes the hydrolysis reaction of a cytidine base pair to a uridine base pair with the loss of an amino group. This editing ability of these cytidine deaminases allow them to perform a variety of essential physiological functions. However, APOBEC, specifically APOBEC3B, is overexpressed in many cancer types, resulting in a high mutational load due to the high incidence of C to U conversion. Some of the essential physiological functions of APOBEC include viral restriction, retrotransposon inhibition, and the removal of foreign DNA in order to protect our body. This pathway represents a potential pathway by which APOBEC may restrict viral infection. During viral infection, cytokines trigger the expression of APOBEC. Then APOBEC damages the viral genome by deamination, while also enhancing the creation of high-affinity antibodies that will help more efficiently get rid of the viral pathogen from our bodies. However, APOBEC may also have off-target effects that damage cellular DNA, leading to DNA strand breaks or mutations, which may result in genomic instability. Genomic instability may then lead to cell death or cancer development and progression. When APOBEC is overexpressed in our bodies respond to the excess of uracil in the genome by a common DNA repair mechanism known as base excision repair, also called BER. BER is initiated when a uracil-specific DNA glycosylase called UNG, otherwise known as UDG, when it recognizes an incorrect base pair and then removes the incorrect base pair, which in this case is uracil. Then, other core BER proteins are recruited to the site, including APE1, Pol beta, and XRCC1, in order to correctly insert the correct base pair, as well as seal the DNA backbone. Interestingly, the accumulation of these uracils by APOBEC3B can be toxic if not removed by UNG. Therefore, we hypothesize that the overexpression of APOBEC3B in cancer cells depends on the expression of core BER proteins, including UNG, Pol beta, and XRCC1, in order to prevent cell death. This is important because these core BER proteins may be potential therapeutic targets in order to selectively kill cancer cells that overexpress APOBEC3B. The first step in our approach was to analyze the endogenous APOBEC3B levels of common cancer cell lines used in the Sobel lab. The cancer cell lines that we analyzed were U2OS cells, which is an osteosarcoma cell line, and glioblastoma cells, which is and LN428 cells, which is a glioblastoma cell line. Nuclear cell lysates of parental U2OS and LN428 cells were loaded on a gel and analyzed via Immunoblab. The results clearly show that U2OS cells overexpress APOBEC3B endogenously, while LN428 cells have very low levels of APOBEC3B. Knowing this, we moved on to the second part of our approach, which included evaluating the role of UNG in cancer cells with high and low APOBEC3B levels using a common system, CRISPR-Cas9, to knock it out. It's already found in literature that the removal of uracil from DNA in cancer cells overexpressing APOBEC3B requires the DNA glycosylase UNG. In order to test this, lentivirus was prepared with 2 CRISPR-Cas9 UNG knockouts with different guide RNA sequences. Then, LN428 and U2OS cells were transduced with the lentivirus and selected with hygromycin to enrich for the knockout cells. Immunobot analysis then determined the efficiency of UNG knockout in the cancer cells. The CRISPR-Cas9 system was used to knock out UNG with 2 different guide RNA sequences that we designed. Guide RNA sequences, also called gRNA, find and then bind to specific desired sequences of DNA, while also recruiting the CRISPR-Cas9 complex to that sequence of DNA. Then, the Cas9 part of the complex acts as molecular scissors and cuts the 2 strands of DNA so that DNA can be inserted or it can be removed as desired. Once we received our UNG knockouts, we were ready for lentivirus production and human cell transduction. Lentivirus was produced with the co-transfection of several packaging vectors as well as one of the desired plasmids into 293F T cells. The 293F T cells were incubated for 48 hours, and then the viral particles were collected and filtered in order to be transduced into U2OS and LN428 cells. Upon transduction into U2OS and LN428 cells, the cells were allowed to grow and proliferate before they were selected with hygromycin in order to select for the knockout cells. In order to test the efficiency of the UNG knockout in these cancer cell lines, whole cell protein lysates were collected and loaded onto a gel. The results clearly show that one of the 2 guide RNA sequences efficiently knocked out UNG, as you can see from the knockout 2. Another interesting result that came from this is that upon selection with hygromycin, LN428 UNG knockout cells proliferated normally, and within a few weeks we were able to collect cell lysates and the remaining cells were frozen. However, when U2OS UNG knockout cells were selected with hygromycin, the cells did not proliferate, which means they did not grow, even after incubation for several weeks. Even then, a very small lysate was collected for analysis. No cells survived for storage. Based on these observations, it was determined that UNG knockout in cancer cells overexpressing APOBEC3B, which in this case were the U2OS cells, is a lethal phenotype. And this is also supported by a recent study by Sarah Brennick and others. Because our knockout cells were not viable for further experimentation, we were forced to focus on a new approach. Our new approach focused on the development of a regulated expression system to selectively overexpress APOBEC3B and LN428 and U2OS cells. In order to do this, a TET-inducible system was created using the gateway LR reaction to transfer the APOBEC3B, its inactive mutant APOBEC3B E255A, and control EGFP open reading frames separately to a lentiviral vector. This allowed selective expression of the open reading frames with the addition of doxycycline once LN428 and U2S cells were transduced with the lentivirus. In order to create the inactive mutant for APOBEC3B, the catalytic residue of APOBEC3B, glutamate 255, was rendered inactive by a mutation to alanine. As you can see here, this is the active residue glutamate, and then it is mutated to alanine. The first step in this approach was expression vector cloning using the LR reaction. The Gateway LR reaction is a recombinatorial cloning system that recombines the @L sites of the entry clone, which contain the desired gene of interest, with @R sites of the destination vector in order to create an expression clone that contained our desired gene of interest as well as a toxic byproduct. The advantage of this system is that it occurs all in one tiny tube. In the next steps, when we transform the vectors into bacteria, though, we may have the potential of having several different products in this tube, including the expression clone, as well as the destination vector and the entry clone. Therefore, we use antibiotic screening protocols in order to isolate just the expression clone. We will utilize the kanamycin resistance of the entry clone, as well as the chloramphenicol resistance of the destination vector in order to specifically isolate just the expression clone that has our gene of interest. We perform 3 LR reactions with entry vector APOBEC3B wild type, the APOBEC3B mutant, and the control EGFP. The Gateway LR reaction transferred the open reading frames from each of these entry vectors into the destination vector PDG2I Puro, which is commonly used in the Sable lab. The desired product of each LR reaction is below. The next step after the LR reaction is bacterial transformation. 1 microliter of each reaction was transformed into stable 3 chemically competent E. coli and then heat shocked for 30 seconds. The purpose of the heat shock is to make the bacterial membrane permeable to the plasmid. Once the transformed cell was transformed, it was shaken in SOC medium for 1 hour and then plated onto ampicillin plates and incubated overnight. The next morning, the plates had several colonies, and 4 colonies were picked for each reaction, and then shaken again in antibiotic-free LB media so that the bacteria can continue to grow. Once the bacteria had grown for 1 hour, they were plated onto chloramphenicol and kanamycin plates to test for entry vector or destination vector contamination as described in the previous slides. If colonies grew on the chloramphenicol plate, it showed us that the bacteria had destination vector contamination. If colonies grew on the kanamycin plate, for example, it showed us that the bacteria had entry vector contamination. These pictures show the results of the kanamycin and chloramphenicol tests. As you can see from the APOBEC3B mutant, the kanamycin plate has no growth on it, meaning that this has no entry vector contamination. While on the other hand, the EGFP, you can see that 3 of the 4 clones have entry vector contamination as seen by the growth on the kanamycin plate. So the positive clones for each vector were identified and they were prepped using Keogen Mini-Prep. Keogen Mini-Prep is a process in which we isolate the plasmids from the bacteria. Once the plasmids were isolated, the concentrations were measured using the NanoDrop 2000 spectrophotometer. After measuring the concentrations of our plasmids, we were ready for lentivirus production and human cell transduction again. And for this, we used the same process as before, in that we used packaging vectors and one of the plasmids, they were transfected into 293 FT cells. The 293 FT cells were incubated for 48 hours before collecting the viral particles and transducing them into U2OS and LN428 cells. Except this time, once the U2OS and LN428 cells were transduced, they were selected with puromycin. Upon transduction of U2OS and LN428 cells and then selection with puromycin, we observed cell death of the U2OS and LN428 APOBEC3B wild type. However, the APOBEC3B mutant and the control EGFP proliferated normally, and after 5 to 7 days, they were able to be split for dox on and off for 24 hours. Following the 24-hour time course, freeze backs of both cell lines were made, as well as whole cell lysates were collected. The TED-ON system is what allows us to create a regulated expression system. For this to happen, when doxycycline is added, it binds to a 3G transactivator, shown here, which then binds to the promoter region, which then initiates transcription of the gene. In our case, when we add doxycycline to the cells, it will activate EGFP and APOBEC3B mutant. And these are the results shown here after 24 hours of the U2OS TET EGFP cells. The green cells are activated with doxycycline. When expressed, EGFP fluoresces a bright green color. So the green cells show us that the system worked great because the doxycycline activated the EGFP. On the other hand, when doxycycline was not added to the EGFP, the cells were not activated, and we also see no leakage in the system because we see no green fluorescent cells. In order to further test the efficiency of our system, we collected whole cell protein lysates of the U2OS and LN428 cells and then analyzed them via immunoblot. We probed for APOBEC primary antibody in the APOBEC3B mutant cells and EGFP primary antibody in the EGFP cells. And our results clearly show that upon addition of doxycycline, that APOBEC3B mutant, APOBEC3B, as well as the EGFP were selectively overexpressed. When doxycycline was not added, they were not expressed, which means our system was successful. In order to try to create a TET-inducible system for the APOBEC3B wild type, we went through the whole process again. We started with LR reaction, and then bacterial transformation, and then lentivirus production and human cell transduction with the APOBEC3B wild type entry vector. And again, upon transduction of the APOBEC3B wild type into U2OS and LN428 cells, we observed cell death. At this point, the circumstances surrounding the COVID-19 pandemic prevented further experimentation so that we could successfully create an inducible system with APOBEC3B wild type vector. Therefore, future work will aim to successfully create the TET-inducible system with the APOBEC3B wild type vector. It will also aim to transduce U2OS and LN428 cells with the TET-inducible systems for APOBEC3B wild type or its inactive mutant with UNG, XRCC1, and Pol beta knockouts separately. The importance of this is that if we can identify the role of these core BER proteins in the overexpression of APOBEC3B, we may be able to identify potential therapeutic targets to selectively kill cancer cells that overexpress APOBEC3B. I would like to thank Dr. Sobel and Dr. Clark for their instruction, guidance, and constant encouragement. Thank you.
Once again, nice work. Thank you so much, Bailey. Throughout your
Great job.
Yeah, really outstanding. I think these two presentations really illustrate something important about the Honors College, as does this whole event. We all know, most of you here at least know, that we've found ourselves on community. We care about each other. We come together for things, even when we have to do it virtually during a pandemic. It's community for sure. But Bailey and Christian have both illustrated to us that important creation of knowledge and culture that Dr. Marshall highlighted, but also leadership. Christian in particular is the leader of our Honors College Association, although of course he's graduating, so we'll move on to our next leader, but has expressed so much leadership. And Bailey is a departmental honor student, so she's joined us more recently, but she also illustrates something really important and interesting about honors, and that's incredible intellectual curiosity. Christian has that as well, but Bailey, I'd like to point out, Bailey, you'll have to correct me on the chat if I'm wrong, but she's actually a music major. So if you didn't know that, there's incredible range and intellectual curiosity that's illustrated there. So I'm just so proud of the students that we work with here. And Bailey, yes, you're welcome. And she confirmed it via chat. So again, throw out your chats. If you have any questions, feel free to throw them out there. Even if we don't get to answer them right here, I think the level of engagement is really nice for the people who made their presentations. And we'll be able to address them at a later point as well. So it's just nice to see your voices expressed in the chat room. We're going to continue now to the next phase of the Senior Showcase. And this is, if you were in the room at the time, we would go from having some food. So, you know, nobody has their picture up, so you can go ahead and nosh a little bit and have your little snacks if you would like while we're sitting here. But we're moving to the phase where we would normally give you your hoods and your medallions. So if you have your hoods and medallions with you, feel free to get them out and sort of join in. And we're going to have a presentation where you're going to hear some kind words from your mentors and see some pictures past and present. So with that, producer Alan, please. Abdullah Alazmi.
Abdullah, although it took me 6 months and lots of pestering from your peers, I'm very happy that you decided to participate in undergraduate research in my group. It's been wonderful watching you find out that you really love research and want to pursue it in graduate school. Your excellent academic reputation translated perfectly into your research skills and your ability to learn and adapt in the lab. There are few undergraduate students who can start a project in the fall and have meaningful and impactful results by early spring. And the fact that you did that is a testament to both your intellect and your diligence. You're going to make an excellent graduate researcher, and I look forward to seeing where your doctoral program takes you. Congratulations, Abdullah.
Prisha Amin.
Krisha Amin, I am taking Dean's privilege so I can brag about you. This is because we've traveled so much together and worked together on research and applications. And I'm so proud of you and so grateful to be able to work with you and looking forward to seeing what you accomplish. My experience tells me that your intellectual curiosity is going to drive you forward in whatever you choose to pursue. I've witnessed it when we were in Memphis and you gave your paper presentation, but also enthusiastically endorsed the idea of walking across the Mississippi with the group, even though a storm was threatening over that big bridge. And going to Oxford, where you, in addition to joining the group and spurring some of the fun evening activities, you also led the workshop presentation that was so powerful. And then in the fall, we worked together. I helped, and you took feedback with such good grace. My many, many, many comments, I know, and created such fantastic applications. I'm just so grateful to work with you and looking forward to seeing what you accomplish. Congratulations.
Sarah Boone.
Hi, this video is for Sarah Boone. Hi Sarah, it was a great experience for me to work with you on your undergraduate honors thesis. I really respect that you challenged yourself to conduct original research using qualitative and quantitative methodologies, particularly when you're a creative. I know working on Due South is a lot more fun than writing surveys and collecting data, but you did a great job and created a very nice study. Congratulations.
Dalton Bradley.
Hi Dalton, it's Dr. Lewis. I'm so glad that I was able to mentor you through your final thesis project, and I think it has been really good for both of us. I think you were good for me in a nice low-key personality that I'm not really used to working with, and it took a lot, but I'm really, really proud of how you're going and where you've gone, and that you were never like, Dr. Lewis, that's just a really, really crazy idea. I look forward to your work, and I'm really proud of you for being very passionate about working with the patients and the patient community and getting your research done through working with a community partner. And I look forward to seeing where you grow as a nurse.
Lauren Casanova.
My warmest greetings to Lauren Casanova, her friends, family members, and the faculty and staff at the Honors College. My name is David Chang, and I'm Lauren's research mentor. For the last 2 years, Lauren has accomplished many achievements, and I want to share a few of those with you. Lauren was inducted into the Lambda Beta Honor Society, and she was also selected to receive the Chairman's Award for Clinical Excellence. And most of all, I think the most memorable moment happened in 2019. Lauren presented a research abstract at the AARC International Congress in New Orleans. And the research abstract moderator was Dean Haas. And Dean Haas is one of the most prolific researchers in respiratory care. And he is one of the most well-respected educators in respiratory care. And I'm so glad that Lauren had a chance to meet with him in person. Again, congratulations, Lauren, and I wish you the best of luck, and I look forward to hearing more good things from you in the future.
Becca Cheatham.
Hi, I just wanted to congratulate Miss Rebecca Cheatham for her honors work here. Even though she's not completely finished yet, she has a little bit more to do. She's been fantastic to work with. I've really enjoyed working with her on a topic that quite legitimately I had very little knowledge of beforehand. And thanks to her and her amazing work from both last summer and all the way through this year, I know personally I've learned a lot, and I know she's learned a lot also. And so I look forward to seeing what she does in the future and wish her all the best from here on out.
Nawel Crinigan.
Well, I've so enjoyed working with you and watching you grow over the past couple of years. I remember the day we met in an advising appointment when you told me of your intent to earn a degree in chemical engineering and with university honors in just 3 years. And as you will recall, I tried to talk you out of it, but unsuccessfully. So I had to agree to support you through the challenge. And I think you'll agree now that it was a challenge, but you were so up for it. With your strong work ethic, your focus, and with efficiency, you have done it. In the lab, your experimental troubleshooting skills are outstanding, and they're going to be missed. But all those skills and your professional manner are going to serve you well in your engineering career. Best of luck to you, Nawel, and congratulations.
Nell Cummings.
Hello, my name is Josh Eisen from Psychology. I want to congratulate Nell Cummings on completing her honors project and thesis. Nell, you developed a really nice survey to measure substance use. You had a lot of data to go through and crunch. I am impressed by your analytical skills and writing ability. It is amazing how many versions of your thesis you have sent. Our longest email thread is 83 messages, which is probably a record for me. You have been a key part of my research lab over the past 2 years. Now, I may have the world behind me, but you have a bright future and the whole world ahead of you. I'm very proud of what you have accomplished, and I wish you all the best.
Sydney Davis.
Hello, I'm Dr. Todd Andel, undergraduate honors mentor from Sydney Davis. I would like to congratulate Sydney on her outstanding work entitled enhancing system security using dynamic hardware. Sydney did some remarkable work, and I wish her the best in her future. I guess this is a little self-serving on my behalf, since Sydney is staying at South to continue her research at the master's level, and she's even keeping me as her advisor. I look forward to our continued progress towards developing hardware-based security mechanisms to combat our ever-changing computer threat landscape. Congratulations, Sydney.
Long Ding.
Hello, Long. I just want to say that when you first approached my research group in March 2018, and you were a biology major, I was quite hesitant to take you based on my previous experiences. But within 2 months, I knew there was something inside you, and when you switched to chemistry, and over the last 2 years, I've seen tremendous growth in your research. You have totally and completely have revolutionized my research projects, several projects. There's nothing that you can't do, Long, and you've proven that you've taken every challenge with superb dedication and persistence. And I know you will be very successful when you go to The Ohio State University, where I was produced. I want to wish you the best, and I know you will have a superb career ahead of you. Take care and best of luck.
Candace Fairchild.
Candace, just wanted you to know how proud we are of you and all the work that you've done, and we know that you're going to do exceptional things in the future, and we're just grateful to have had you as a student. So keep up the good work, looking forward to the fall, and just darn proud of you, okay?
Nicole Green.
Hi, Nicole, it's Dr. Lewis. I'm just wanted to say real quick how proud I am of you for persevering. I know there was times that you just wanted to give up honors because you're just like, I don't know if this is for me. And I'm glad you stuck with it. I'm glad you followed my advice, whether you thought it was kind of crazy or not. And I'm really glad that we were able to get our Zoom experience well before the pandemic hit. I think you're a pretty big Zoom champion right now and a little zoomed out. So I'm so proud of you, and I look forward to seeing where you go as a nurse.
Jordan Harbison.
Jordan, I am so very proud of you for all the work that you've accomplished while working on your honors thesis. Starting last spring, working through the SURF project over the summer, and then in the fall and this spring. I'm especially impressed with how well you've mastered the complex modeling aspects of your project, in addition to the difficulties of working with supercritical fluids at high pressure experimentally. Both these skills are going to serve you well as you start your career as an engineer. It has been an absolute pleasure to work with you as a student in class and as a researcher. You're going to be an excellent engineer and a value to the company that you work for. Again, congratulations.
Aubrey Harville.
Congratulations, Aubrey, for making it this far in the University of South Alabama's Radiologic Science program. Also, congratulations are in order for completing the requirements for graduation from the University of South Alabama's Honors College. As chair of the Department of Radiologic Sciences, I do believe that the project that you have chosen as your thesis will make a difference in our profession. Anxiety in pediatric patients that come to the radiology department has always been a challenge for the technologists. Completing an examination and providing quality diagnostic images to the radiologists expedites diagnosis in these patients, which results in better patient outcomes. Educational field trips, as you have defined to this population, of what to expect when they arrive for their examination will hopefully reduce the fear of the unknown. It has been a pleasure to work with you on this project. Your professionalism and your desire to do a good job make it easy to be a mentor to you. Good luck on your future endeavors, and I hope that life brings to you all that you are looking for.
Savannah Howie.
Wow. Savannah, as you can tell, this is a very important video since I broke out my suit. Well, at least half of it. I want to congratulate you on all of your accomplishments at South, including, but certainly not limited to, your academic achievements, all of your research achievements, service to multiple student organizations, your multiple internships, and if that's not enough, as a pole vaulter, you use your spare time to jump over bars that I would need a ladder to reach. Many students strive to accomplish only a small amount of everything you have. This is a testament to your drive and determination. That's why when you started looking at graduate schools, it was my pleasure to write reference letters and talk you up to my colleagues. I know you will do very well at Auburn and represent South. Savannah, I am very proud to have you as a member of my research team and an honors graduate of the civil engineering program.
Kristen Waite.
Congratulations, Kristen Hewitt. How wonderful it is to be with you at the virtual Senior Showcase. After watching you go through and persevere through your thesis project over the last year and a half here at the University of South Alabama, when you first came in to ISC 245 back in the fall of 2016, I bet you never thought that that would lead you to listening to, coding, and analyzing hundreds of Spotify's top charting songs over the last 3 years. I appreciate your dedication to the difficult and time-consuming project and detailed analysis that you undertook. I look forward to hearing how you merge your data analytics skills with your future endeavors. Congratulations, Kristen.
Zara Ijaz.
Hey, hi, Jazz. So, I'm super proud of you. I hope to see you around the med school this next year. I'm sure I will. You're always welcome to come and do a project in my lab if you want to work. Continue working with Salmonella. I'm hopeful we'll get a grant this year and move forward in large part that's due to some of the work that you've done. Congratulations on med school. I'm very proud of you.
Abigail Jernigan.
Abby, it's been a delight to work with you over these 3 years. I can still recall how curious you were during sophomore year when you first approached me about our research in hydrogen generation using sunlight and photocatalysts. It's such a pleasure to see the passion that you bring to your work, drawing connections from your love of beauty to your commitment to environmental conservation and onto your aspirations towards a career in energy research. You seem so confident when you talk about it, and that confidence makes you ready to embrace new opportunities, like last year when you went off to Poland, and now you're off to Colorado School of Mines. I'm really proud of you, and I look forward to seeing you harness your passion as you pursue this new adventure. Congratulations, Abby.
Kiara Joyner.
This presentation is dedicated to my student, Kiara Joyner. This is Dr. Mata. You worked with me for the past year and a half on the genus Amanita. Here you see your first collection that took you about 2 hours to complete and describe. We later went in May for a mushroom walk around campus where you collected several more, and you started to become more and more acquainted with all the hard work that you had to put in later on. I took you in July to the summer foray by the Gulf States Mycological Society, where you were immersed into lots of different taxa in this particular genus that is so awesome to look at. You also had the opportunity to talk and interact with the president of the society, David Lewis, who is one of the authorities in mushrooms in the Gulf Coast and in general in North America. So you really had this wonderful opportunity to work with bright people that know more mycology than both of us know. You also went with me in September in 2019 on my annual mushroom walk that I do in Crosby Arboretum, where you collected even some more specimens, and I think you have a couple of those in your thesis. And we then later collected more in August or September and found this precious and beautiful mushroom, Amanita arkansana, who I think is a new report for Alabama, and we just need to make sure. So good for you. This is an also awesome find that you did and were able to process in your thesis. As a result of your hard work, Chiara, not only you presented in the SURF Symposium, but also you were invited again to give an oral presentation at the Winter Foray in the Gulf States Mycological Society in Louisiana. And then later on, you participated in a much broader meeting by the Mississippi Academy of Sciences. So I think you have done a wonderful job, Chiara. I am really, really happy that you went the extra length and extra hard work. I know I have been tough on you with your corrections in the thesis, but that is going to pay off. 45 pages of writing is a lot, and I think you have done a fantastic job. There is not an image of you and me alone, except for these students with Eric Summer on that presentation of SURF Symposium in 2019. So with that, I want to thank you again for your work, Chiara. You did fantastic, and I wish you only the best in the future for you.
Sydney Jones.
Hi, my name is Nicole Amari, and I'm here to offer praises about Sydney Jones' honors thesis project. She is a student in the English department, and her project on empowering ESL student learners through flipped grammar instruction to help improve their writing quality has been a great project. She did an independent project. She secured IRB approval in order to do a pre-test and a post-test of ESL students at a community language instruction center, as well as the English Language Center here at University of South Alabama. What most impressed me about this project was all the risks that Sydney took. It would have been very easy to be safe and do things that were easy, but I saw her time and time again try to ask some hard questions and take some risk in her data collection, including doing extra in terms of analysis and focus groups. And she learned very quickly that data analysis is not always easy and smooth, but she ended up with a great set and some excellent analysis of lexical and syntactical complexity of these student writers. And it was a fantastic project, and I really enjoyed working with her. And I know she's going to do awesome in graduate school with whatever she pursues. Really had the pleasure of working with her. Thank you.
Caroline Jordan.
Hi, Caroline. Congrats on wrapping up an excellent undergraduate career here at South. It's been a pleasure working with you in class and in lab for the last 2 years. I knew you were a keeper when you and your team showed up in these t-shirts for your end of semester presentations in our genomics class, and the work you've done since then only confirms that assessment. You've done outstanding work on 2 different research projects over the last 2 years. The first building on our course-based research in genomics, which you and Lisa presented earlier this year. The second working on the characterization of these antimicrobial producing bacteria. It's been a strange spring, but I'm looking forward to getting together in person to wrap things up once we return to normal. Congratulations again.
Emma Kalani.
Hi Emma, it's Dr. Lewis. I am just so proud of you for all that you have persevered through. I know the spring of 2020 was not what you expected for your senior year, but I'm so glad I got to know you over the past few years, and I will never forget you walking into my office saying, hey, I made up this fake research project, can we do it? And then I was like, no, that's actually a real research project, let's get through it. And I'm so proud that you did, and I look forward to publishing your research with you. And I know you're going to be the best palliative care nurse and progressive care unit nurse. And I look forward to watching your adventures.
Naden Kreitz.
Naden, congratulations. When we started all of this, I wasn't so sure that either of us was going to make it. You changed topics, entire subject areas, endured numerous drives to the most rural of Alabama, dealt with some difficult people, survived my brutal editing, and even stayed calm when an unprecedented global pandemic derailed our entire project. You are by far the most persistent, tenacious student I have ever had, and that is going to be one of your greatest assets. No matter what, I had no doubt that you would get the job done, which I can't say of everyone. You are smart, a very hard worker, and you brought a lot of fun and levity to our team. You're going to go far in life, and it's going to be great to watch. You know you can always depend on me as your mentor and even as a friend going forward. Keep making me and everyone around you proud with your countless achievements to come.
Fernando Lorenzo.
Hi, I worked with Fernando Lorenzo over the past couple of years actually, where he was doing a senior honors thesis looking at alkane solubility in lipid-inspired ionic liquids using molecular simulations. Lorenzo is an electrical engineering student doing a chemical engineering themed project, but he never seemed out of his element with us. I think what I enjoyed the most about Lorenzo was that he was always looking for something to do, which is probably the greatest trait you can have. And I'm so fortunate that we had this opportunity to work on this project together. It worked out better than I could have ever imagined at the beginning. So anyway, very proud of the work that you did, and best of luck next year.
Elvira Mando.
Really lucky to have worked with him over the past couple of years. His enthusiasm for programming made it a real pleasure to work with. I'm so glad we got to connect on that level. Very proud of all of the work that he's accomplished, and I look forward to his continued success at Georgia Tech next year. Best of luck.
Christian Manganti.
For the last 4 years, it's been a true privilege working with Christian Manganti here in my lab at the Mitchell Cancer Institute. He has worked in several different capacities at MCI, and most notably spent 2 years working on his senior honors thesis project, which I'm very proud of, and hope that he will be able to continue as he begins studying at the College of Medicine here at USA in the fall. He's welcome back if he can find the time, which I think he will. He's a master at time management and juggling many balls at once, always with a strong positive attitude and just a really genuinely great guy to be around. Christian, you deserve all the success, and I wish you all the best that life can bring you in the future. Congratulations.
Bailey Manning.
Good evening to all of you attending the USA Honors College Virtual Showcase. My name is Robert Sobel, and I am a professor in the Department of Pharmacology and the head of the Molecular and Metabolic Oncology program at the Mitchell Cancer Institute. It is my sincere pleasure to say a few words about one of tonight's USA Honors College students, Miss Bailey Manning. I actually came to work with Bailey by accident. Her chosen summer undergraduate research fellowship lab could not participate in the SURF program last summer, and so I was asked to help out. She then, of course, continued with us all year for this honors program. This was a stroke of luck for us, as Bailey was outstanding. I'd assumed then that Bailey must be a superstar in the biological sciences program, but was shocked to learn that she was in fact a music major specializing in piano performance. But as a pre-med student, she was up to the task and completed a cutting-edge project. We are all lucky that she will continue to be part of the USA family as she attends med school here in the fall. Best of luck, Bailey. We are all proud of you.
Samantha Mays.
Hello, my name is Dr. Leavesley. I'm a professor in chemical and biomolecular engineering, and it is my great pleasure to introduce Mrs. Samantha Mays for her introduction into the honors colloquium. She was a departmental honors student in our department, and she's just done a really outstanding job during the time she's here. So instead of tell you a lot about it, I thought I would maybe show you a few things that she's done, because I'm just very impressed with the level of work, the quality of work, and the level of understanding she's achieved. Samantha went through developing a very complex set of Monte Carlo based optical ray trace simulations for us. I won't explain what they all are, we don't have time to discuss that, but I will show you that she dealt with a lot of different, again, very complex properties of the optics. She went through modeling entire systems with ray trace modeling. You can see rays shown here in red, collecting all of the output information, optimizing the model. But then she went really a step forward and developed this 3D simulation in CAD software, worked with manufacturers to prototype the pieces, assembled all of it, designed custom circuit boards and other components, and then finally tested the device. And I'll leave you with just a short video showing how this runs. It's really quite impressive. As you can see, there's a set of different wavelengths of light that are all coupled into a common liquid light guide output that goes into a microscope or an endoscope and lets us illuminate samples with many different wavelengths for looking at the molecular properties of different cells and tissues. So I just want to say, Samantha, great job well done. Thank you for all of your hard work and congratulations.
Evan Mazur.
Hi everyone, I'm Dr. Stephanie Smalligan, and I'm an assistant professor here in the Civil, Coastal, and Environmental Engineering Department. I've had the pleasure to work with Evan Mazur, who completed her research on coastal engineering, which is what my specialty is, and she was able to study hurricanes through numerical modeling and present her work at national conferences. I am so impressed with the progress that Evan's made throughout her undergraduate career, and I'm so thankful that she chose to work with me on her project. So to Evan, congratulations. I'm so proud of you, and I'm looking forward to one day very soon to call you a colleague.
Shuba McClelland.
Congratulations, Shuba, on this milestone, so completing your undergraduate degree, but also your thesis. From the beginning, you were the easiest student to mentor. I could give you just a few things to do with very little detail, and you would go for a week or two and work on it and come back with a product of high quality, with even more questions that we could talk about. You really have a knack for research. Something else that was just surprising to me was how easily you worked with our research participants who were communicatively impaired, and you just kind of naturally were able to interact with them, which I thought was just amazing. So I'm excited for your future. I think everything that you do is going to be amazing and great, and wanted you to know that I really enjoyed mentoring you this year. I hope that you learned a lot through the process, and I can't wait for you and to see what you're going to do in the future. Congratulations.
Ian McCullough.
Dear Ian, it's been a pleasure to have been your instructor and research mentor over the course of the last 2 years. You joined my lab in the beginning of your junior year, and this was the period of time that I needed a student to test some ideas. You came in, learned the techniques quickly, got some data, and this earned you a co-authorship. I've been extremely pleased with how you've taken ownership of your thesis project, and you have developed into a wonderful writer. As you move on to a career in medicine, I hope that we will still be able to keep in touch as we need to put this paper together. Again, Ian, congratulations on a job well done, and I wish you much success.
Avery McNeil.
Good
It has truly been a pleasure working with Avery McNeil over the last 18 months. Avery is an exceptionally bright and hardworking young man who has worked on a very challenging project and persevered through numerous setbacks. I am proud that Avery is a recipient of the prestigious Graduate Council Fellowship at the University of Alabama. I wish him all the best in his future endeavors, and I hope that we will continue to work on mathematics together. On a personal level, I've really enjoyed getting to know Avery. He is a very soft-spoken and kind young man. He has put up with my sometimes hectic lectures, and more importantly, even laughed at my corny jokes and dealt with various cat-induced shenanigans during our Zoom meetings. He has been my first advisee, and I consider myself lucky to have had him as my first student. Good luck in the future, Avery.
Lindsay Meyer.
Lindsay, congratulations on finishing your degree. It was great fun working with you on your honors project, and we have lots of presentations and publications that will come out of it. I wish you the best of luck as you go on to this next part of your career. And also remember that we will miss you at Passage USA. Feel free to come and visit us at any time.
Joseph Milter.
Joe, your enthusiasm for learning was evident from the first time you asked a question in my class, and it carried over 100% into your research. You had a challenging project of taking a successful research idea and making it work backwards so that it was industrially viable, and you made great progress in a very short period of time. You've also demonstrated an excellent ability to communicate scientific information, giving presentations and posters. And this was recognized by your award for your poster presentation at the ASCHE National Meeting. I am very proud of you and your accomplishments, and I'm sure that you're going to excel at your doctoral program at Notre Dame. Never lose your enthusiasm and love of learning. Again, congratulations.
Apurva Nagaraj.
Hi, I'm Jack Shelley Tremblay, the chair of the Department of Psychology, and I'm very proud to have worked as the mentor for Apurva Nagaraj, who is a graduating senior and member of the University Honors College. She's graduating with her degree in biomedical sciences, and I've been working with Apurva for the last 2.5 years, and it's been a great experience. She has worked so hard, and she has brought in students with learning disabilities and their families from the community, brought them into our lab, learned a tremendous amount about reading and language and about the visual system and about how the brain works, and she's put together an outstanding product. We're very excited for Apurva, who wants to go into the field of healthcare, and I expect great things from her going forward in the future. So congratulations, Apurva.
Ajay Padada.
Hello Edja, congratulations for your achievements and hard works. I understand and share your passion for machine learning. You overpassed AI capabilities in many ways: creativity, manipulation of complex concepts, unique human skills, and ability to be systematically late. I really wish you good luck with your future training and hope to read soon your research articles. Again, many thanks from the Department of Neurology.
Josiah Perry.
Josiah, I don't think I've ever seen a thesis face so many obstacles. There's so many things that are beyond your control. I'm not just talking about the pandemic. There are many other factors that really made this task difficult for you. So I have to say I admire your initiative, your perseverance, your patience. Your resilience has just been amazing, and I wanted to thank you and commend you for all your good work.
Jesse Puckett.
Jesse, I am so proud of you. I wasn't sure what to expect at first since you were in the Misfit Toys section of Honors 301 that I was teaching, and you were so laid back compared to my complete and utter opposite of being laid back. But I'm so happy that we rolled the dice on each other. You are so hardworking and dedicated. You were even willing to take on basically an entire second project with no questions asked, and you were instrumental in completing it. You took everything in stride, you helped balance my anxiety, and you were truly a pleasure to work with. You understood the work that we were doing, which isn't actually true for all students. So ultimately, I feel like we were both very successful. Remember, you always have me to rely on as a mentor and as a friend. Now go take on the world with passion, poise, and purpose.
Lindsay Roden.
Hi, this is Dr. Lindsay. I would like to congratulate Lindsay Roden on the completion of her thesis. And also, Lindsay, I just want to give you a shout out to say how very proud I am of you and all of the hard work that you did during the time that you were in school. We are especially proud in the College of Nursing that you completed the summer SURF and that you also were accepted to present your thesis at the Alabama State Nurses Association's conference in April. It's so unfortunate that the pandemic hit and you were unable to present it, but we are very proud of you and feel like that the work that you've done is going to be super important to maternal child nursing. So thank you so much for all of your hard work and best wishes on a wonderful.
Jeremy Shiver.
Jeremy, congratulations on your graduation. I'm going to miss our Friday afternoon meetings of going through data and trying to figure out what it all means. I wish you the best of luck in your future. It's been a great experience. Please keep.
Ashlyn Stalley.
Hey Lee, very proud of you. Done an awesome job. You know that. Your project's going super well. I imagine we'll have a paper published this year. But I couldn't be more proud of you for your decision to pursue an MD PhD. And I think Colorado's where you're supposed to be. So Anschutz is an excellent institution, and they're lucky to have you. Again, couldn't be prouder of you. Great job, Staley. Keep me informed as to what.
Andrew Theodore.
Hi, it's Neil Schwartz, and my mentee was Andrew Theodore. And I enjoyed working with Andrew very much. We were able to collect data for multiple studies, one looking at acute epicatechin supplementation on blood flow, and also the effect of inhaling caffeine on performance, mental performance, and physical performance. But Andrew was great to work with, got along real well. Like to joke around a lot, but I wish him the best of luck. UAB Physical Therapy School, gonna miss him. Wish he'd stay here, but he'll be a great physical therapist, and I wish him well and hope to follow his career throughout the future. So good luck, Andrew, and congratulations.
Lydia Turner.
My name is Michelle Strong, and I am an associate professor in the Department of History. It is my very great pleasure to introduce you to Lydia Turner and to congratulate Lydia for her accomplishments in the honors program and at USA more broadly. Her honors thesis on environmental justice, sustainability, and the textile trades in 19th and early 20th century Britain stems out of a concern for class inequalities that began, I suspect, long before she learned about the Industrial Revolution, or for that matter, Jeremy Bentham. Lydia is fast becoming a promising young scholar who will have important contributions to make in how we understand the world, past and present. Lydia, when I think of you down the road, I won't need an emoji to spark a memory, although if I did, it would be a happy face, of course. It has been an absolute delight and a privilege to work with you. Good luck in all your future endeavors.
Seth Benson, cheering you on. and Allison Mishner cheering you on. So officially, with the acknowledgement here, which Dr. Marshall gave earlier, we want to thank the USA Foundation as well as the Central Administration. Officially, that is the end of the showcase as we planned it. Don't rush off quite yet. First of all, I think Alan, Dr. Marshall, and Ms. Pam all want to say something. And then we want to encourage you to turn on your video screens and kind of whoop it up. So Alan, you want to say anything a minute?
Sure. I just want to wish my fondest congratulations and how proud I am of you guys and all of the hard work that you've done. And I'm definitely getting misty over here. You guys really mean a lot to me. If you don't come and visit, I'm going to be upset. But no matter where you go and what you do, you are going to bless the lives of everyone that you encounter. Because that's what you do. You are not only incredibly intelligent, but also incredibly hardworking, incredibly giving, and incredibly kind. So take that with you and share it with the rest of the world.
Dr. Marshall?
You're muted. You're muted. Oh, let me see if I can unmute him.
Here. Dr. Marshall, I'm sorry to tell you this, but brilliant things and you're muted.
Yes, every year we end up wondering how we're going to function after this group leaves. And that's always the case. We always manage it somehow. But nevertheless, you guys are really going to be hard to replace. So I'm going to miss you guys a lot. Congratulations, though, and I expect nothing but greatness.
Thanks. Ms. Pam?
Hey, I just want to say I'm so proud of y'all, and I'm going to miss y'all, and y'all will always be my babies. So love y'all. Best wishes. Short and sweet.
And to the point, it's the essential points, all three of you. And then I just want to throw in also how sad I am that we couldn't do this in person. I, like Alan, am a little misty-eyed. That doesn't happen with me a lot, so you're special. Actually, it happens plenty. I shouldn't say that. But we're gonna miss you. And this has really been, I think, a special event. I enjoyed having the mentors directly speak to you through the video, and it made me smile. It made me a little teary-eyed, and I recognized many things in many of the things that were said. So thank you to the mentors for doing that. Before we go too crazy here, I also want to thank Alan. Alan did that last presentation. He pulled together all of the video, all of the layout. Ms. Pam pulled together the photos and helped to shuffle them to Alan, and then made sure all the details got out there. And Dr. Marshall, I'm going to be curious how many of you did your 3D goggles and what you thought. We'd love to have some comments, either using your words or using your chat. But that was Dr. Marshall and Marketing and Communications with lots of brainstorming. And we were really happy to give you a chance to walk around the Bethel. I have to say, only today did I figure out how to do it right. And I almost walked into many things in my house, but I honestly thought I was in the Bethel. So it was something. Anyway, congratulations to you all. Thank you for coming. Turn on your video screens and let's whoop it up a little bit. All right.
I do have one final thing.
Oh, Alan.
Sorry. I will be sending the specific clips to you students. So the clip that your mentor gave us, I will be sending it to you. It may take some time. I'm going to try to have everything uploading while I sleep tonight because it's lots of videos, but I'll try to have those out by tomorrow at the latest Monday.
Good, and this has all been recorded. It might go on Facebook. There's lots of things in the works with marketing people. We're really glad you showed up. Everybody, woo!
Yay!
Make some noise. Feel free to put on your microphones now. I apologize for the names I got wrong under pressure. Okay. Anybody want to say anything? Feel free.
my roommate hooded me, so I'm not going to take it off now.
Yay!
We were thinking about saying to put your video screen on when your name came up and show yourself get hooded, but it seemed like it was too complicated. Awesome. Anyone else want to say something? Thank you for coming.
The VR thing was super cool. Whenever I got it in the mail, I was very confused. I put it together and I was like, they have something planned for this. I'm just gonna set it to the side. But I thought it was really cool, very creative.
Nice.
It took me a while to realize it. It's gonna be something, and I try to like hold it like this.
Yeah.
Finally, until this morning, I got the video, and then like, okay, yeah, it's gotta be it.
Very good, very good. Mentors, you're probably jealous not to have this. We'll work on getting you some as well. But the first priority was to get things in the mail, which is also thanks to Ms. Pam and Dr. Marshall played a big role in that as well.
I have to say, this was the first year I was involved here, and I just am totally impressed with this whole process, and also the level of the work by everybody. I looked at all the posters earlier today, and just fantastic, just really amazing.
Fantastic, thank you. Cool. Well, I hate to hit leave meeting because then it means that the academic year is done, but it's probably time to leave meeting. Ugh. But I do hope we'll get y'all together in person when we can. Imagine something in a year, maybe, maybe sooner, but okay. And in the meantime, please stay in touch.
We're gonna miss y'all.
Yep. We already do.
We already do.
All right.
Yep.
All right. Well, send messages if you have anything. Otherwise, I guess it's time to leave meeting.
Bye.
Bye, guys.
Great to see you all.